Okay, good afternoon, everybody. My name is Bob Bruner. I am the exotic forest pest educator with the Department of Entomology with Purdue University. Today is our second session of the Emerald Ash Borer University. This is a program done in partnership with Michigan State University as well as the Ohio State University. My colleagues on this program are Sheila Babbitt from MSU and Kayla Perry from OSU. And they will probably pop in here in a little bit. Today, we are hosting David Burke, the Vice President for Science and Conservation at Holden Forests and Gardens. And today, he's going to be talking to us about a subject that I have been really looking forward to about beech leaf disease. So, David, the floor is yours.
Great. Thanks a lot, Bob. It's great to be here to talk with all of you today about beech leaf disease, which in some ways is a new disease. It's a new nematode disease. Some of us have been living with it for about 10 years, which seems kind of new. But a lot of people maybe don't know too much about beech leaf disease, especially if you live sort of to the west of Ohio or the south of Ohio. The disease may have not made it into your region yet. It's been spreading mostly to the east. So if you're in the Mid-Atlantic region, or for example, you're in New England, you probably become very well acquainted with beech leaf disease. But it's been around for about 10 years or so. I'm at Holden Forests and Gardens, and we're here in Northeast Ohio. Holden Forests and Gardens is a combination of the Cleveland Botanical Garden and the Holden Arboretum. So I'm based at the Holden Arboretum. We're about 25 miles or so to the east of Cleveland, not far from the shore of Lake Erie. And a lot of the work that we do here is within our forests and natural areas, and also in partnership with the U.S. Forest Service. And the picture I'm showing here on the left is a beech bark disease resistance plantation. This is a plantation of about 100 trees, beech trees, that were developed by Jennifer Cook and the U.S. Forest Service for their resistance to beech bark disease. Unfortunately, they've developed beech leaf disease. But it's a good opportunity for us because we can work with fairly large trees, but in pretty convenient setting. And some of these trees do, as I'll show some resistance perhaps to beech leaf disease. So maybe that's a positive note to start this off on. The picture there on the right is a beech tree that actually shows or displays signs of beech leaf disease, the early kind of stages of the infestation and the disease progression. And you can see the characteristic dark banding between the veins. So you have this dark intervenal banding that is characteristic of the disease early on in the disease development and progression. So what is beech leaf disease? So as I mentioned, we've been dealing with it about 10 years or so. It was first identified in Lake County, Ohio in 2012. And so the Holden Arboretum spans both Lake and Geauga County. It was found in the Lake Metroparks, actually. John Pogachnik was a naturalist there, and he first noticed this disease spreading within some of their natural areas, and then brought the attention of this disease to others in the research community. And so people began kind of looking into it at that point. No one really knew what it was. It showed up here at the Arboretum in about 2014 and began spreading from this area, really the epicenter, to other places in Ohio, Canada, and areas further to the east. As I mentioned, the disease, as you can see on the top image here, is characterized by this sort of dark intervenal banding that you see. As the disease progresses, the leaves tend to become leathery, very much thickened, but also they tend to sometimes shrink in size. So you'll go from that picture you see on the upper right there to leaves, as you see on the lower left, on the lower right, which are also, that's a fairly full branch, but they're becoming leathery and thickened. And then in extreme cases, they begin to actually shrink. And in many cases, the leaves will abort or not open at all during bud break. So you get this progressive browning of the tissue in many cases. Again, this is a little bit more of a close-up of what this looks like, beech leaf disease in the early stages there on the left, with again, that sort of dark intervenal banding that you see here. And this is an example of what we mean by a sort of thickened, leathery kind of leaf, where they become crinkled. And then in extreme cases, you can see here, some of these leaves are beginning to become really deformed and very small in size. And as this disease progresses, you see more and more of those leaves on the right-hand side on the trees and the canopy And as I mentioned, eventually, as the disease progresses, the leaves hardly open. You'll get abortion of the buds, or you'll get very, very small leaves that open up on the tree. And of course, this leads to significant canopy thinning and can eventually lead to tree death. The cause of the disease was not known. A lot of folks got involved with trying to understand this disease, ourselves included. We talked about bacteria, we talked about fungi, but David McCain, who's with the Ohio Department of Agriculture, first noticed nematodes on leaves that had symptoms of beech leaf disease that had been sent to him. And so the path kind of changed and went down this avenue of whether a nematode could potentially be causing this disease. And so a large number of people were involved in that research, ourselves included, and a paper published by Lynn Carter in 2020 identified or confirmed that the nematode Littleanchus crenatae, subspecies McCannii, after Dave McCain, was associated and required for beech leaf disease. And again, you see that dark banding here between the leaves. Now, one of the ways that we identified and showed that this nematode was causing the disease was by actually collecting the nematode from leaves and buds in the field and extracting the nematode from that material. And basically what we do is we take the leaves and the buds and we float them in water. We could be using a Behrman funnel, for example, or you can put them in petri dishes with a little bit of water, and the nematodes will come out. And at that point, we can count them underneath a dissecting scope, a compound microscope usually, at low magnification, and we can count them under the scope. We can estimate how many we have, and then what we do is we actually take those live nematodes and we add them to dormant buds in the greenhouse. And so this tree here was in the greenhouse, did not have beech leaf disease, and we inoculated a bud, which was way down here in the lower left of the picture, with the nematodes in fall. And then basically waited through the winter until they opened up in the spring. And then buds that received the nematode opened in the spring to reveal this characteristic intervenal banding that you see here. So we've done a fair amount of work with this. We have done some other types of experiments where we take the wash water, for example, and we add it to buds, thinking maybe there's a bacteria, fungus associated with the nematode that could cause beech leaf disease. But we never see beech leaf disease unless we actually add the live nematode. And so this was an effort to kind of close Cook's postulates, take what we thought to be the causative organism from symptomatic trees and individuals, isolate it, reintroduce it to asymptomatic individuals under controlled conditions, and we see beech leaf disease develop after that work. So this indicated to us that the nematode was required for beech leaf disease. These are some photos that Lynn Carter and her group at the USDA took with a scanning electron microscope. Some of these photos were taken from leaf tissue, beech scales, bud scales, collected here at the Holden Arboretum in that plantation I mentioned a little bit earlier, the beech bark disease resistant plantation. And what you can see here is you can see the nematode associated with just underneath the bud scales themselves. And then, of course, you can see it here as well, and then here in a much magnified image. And so this is what these nematodes look like within the tissue itself. And then these are also scanning electron microscope images from Lynn Carter and her team, really looking at leaf tissue and what it looks like in the presence or the absence of the nematode. So there on the left, you have sort of that standard anatomy of a leaf with the palisade layer and the mesophyll, and you can see the vascular tissue there. And on the right, you see a cross-section of a leaf that's affected by beech leaf disease. And you can see how disordered the tissue is, how you don't have those distinct layers any longer. The tissue's been damaged. And if you really focus in, what you can see is you can actually see the nematode in and among all of that damaged and disorganized tissue. So again, this is Littleanchus crenatae McCannii, as we're calling it, subspecies McCannii. It was first identified, Littleanchus crenatae, was first identified in about 2018 and was isolated from Japanese beech in Japan. So Kanzaki et al. Published that original paper identifying this as a new species in Japan. And of course, we were dealing with beech leaf disease here already. It's uncertain how it got into the country, but it was identified in Japan in 2018. And then, of course, found to be the causative agent of beech leaf disease, starting with our work in about 2020 or so. So yes, Little Ancus crenata after Japanese beech from which was isolated. Japanese beech is Fagus crenata. So that's what the nematode looks like within tissue. In addition to causing damage to the tissue, some folks, Paulo Vieira with the USDA has investigated some of the cell division that's associated with the nematode. And so what you can see here is you can see the control image there on the top part of the slide. You see a sort of a healthy beech leaf. And again, you have well-organized tissue. You have maybe, maybe 8 cells deep as you go from the top to the bottom of that leaf. But as the nematode infests that leaf and begins to cause damage and begins to feed, then what the nematode is effectively doing is it's dramatically increasing cell division. So as you go from the B image to the C image to the D image, you're having increasing numbers of nematode and increasing symptoms of beech leaf disease. So what the nematode is doing is it's dramatically increasing cell proliferation, cell division. So instead of having, say, 6 to 8 cells across for a control leaf, probably more like 8 to 10, you're getting 40 or 50 cells deep as you go from the top to the bottom of the leaf. And that probably explains the symptom and that you're getting this dark intervadal banding, right? So as those cells proliferate, you're getting that darker and darker color in those places where the nematode is found in between the veins. So it's causing this massive cell proliferation, which again is related to that symptom development. And we see this also with European beech. So we've worked with Paulo looking at European beech as well, and you see the same general patterns with European beech. And this disease affects not just American beech, but other species of beech, including European beech. So as I mentioned, when we're working with the nematode, we can take leaves and buds. We can place them into water, whether it be a Berryman funnel or just a Petri dish, and we can pull those nematodes out and enumerate them in count scope. This is what the nematodes look like through a scope. You can have anywhere from a few hundred to several thousand nematodes within a sample. Typically, we take samples that range from maybe 5 to 10 leaves from an affected tree. And if we're taking 5 to 10 leaves, like I said, we could have anywhere from a few hundred to several thousand nematodes coming out of that tissue and into the water solution, whether it be a Petri dish or whether it be a funnel. And they can be very, very active, obviously. Kind of interesting critters. They are extremely tough. We have taken them and we have placed them, for example, in a -80 freezer to try to kill them. And after an hour, we take them out and many of them actually wake up and start moving around again. So they are tough to kill. I think it's taken us up to 2 hours to kill them in a -80 freezer. So they're tough buggers. Early on, we thought about whether other organisms, whether bacteria and fungi, for example, could be causing beech leaf disease. And certainly we see evidence for changes in bacterial and fungal communities on and associated with leaves. So the microbiome, if you will, of the leaf will change both with symptoms of beech leaf disease and also the nematode itself. But we don't really think that this is a bacterial fungal disease. It's possible that there could be some involvement with bacteria and fungi in the nematode, but we really do think this is a nematode disease. I'm showing an image here, a slide from a paper we published in Applied Environmental Microbiology, where we looked at fungal and bacterial communities associated with symptomatic and asymptomatic tissue across locations in Ohio and Pennsylvania. And we see regional differences in both the bacterial and the fungal communities on leaves. And we see differences in this case, the fungal community between asymptomatic and symptomatic tissue. So highlighting here the presence of Ericife, much more abundant in symptomatic tissue than asymptomatic tissue, for example. And this is a genus that causes things like powdery mildew. It's typically present. We can find it on asymptomatic beech, but a fairly large increase in its relative abundance in symptomatic tissue. So things that, leaves that are symptomatic for beech leaf disease, we do see an increase in that genus, for example. But we also don't see it in symptomatic tissue. So you can notice that there are a number of samples here where we really just don't see it present. So even though it may be increasing in abundance in the presence of beech leaf disease in some locations, in other locations, it tends to be absent. So kind of going along with that idea of Cook's postulates, where the agent of the disease has to be present whenever you have symptoms of disease. And that's just not what we see with many of these fungi and bacteria. We may be seeing shifts, we may be seeing an increase in the relative abundance of some groups, but we don't necessarily see these organisms every time we have beech leaf disease symptoms. But what we do see all the time is the nematode. So the nematode appears to be the causative agent. And that's to say bacteria and fungi couldn't be interacting or causing secondary opportunistic infections, for example, right? So the damage that the nematode might be causing could make the tissue more susceptible to something like powdery mildew. But that's not, this fungus is not what's causing beech leaf disease in this case. Now, one of the interesting things about beech leaf disease is its population sort of cycle. We began doing some work in 2019 in our plantation here. That's the beech bark disease resistance plantation, where every few weeks we collected buds and leaves, buds or leaves, depending upon the time of year, or if both were present, we collected both. And what we did was we used a DNA extraction and quantitative PCR method to look at changes over time in population size. So we're using detection of the ribosomal RNA gene in nematodes as a measure for population changes. So we were using a primer set that was specific for nematodes, allowed us to amplify Littleanchus crenata mccannii in our samples. I'll tell you that, with really several hundred thousands of samples, this primer set when we're working with beech, this primer set gives you back Littleanchus crenata. We don't tend to see other nematodes associated with beech leaves or buds, just Littleanchus crenata mccannii. So we feel pretty confident that this primer set is actually amplifying Littleanchus crenata and nothing else. Although we have developed other primers that could be used under these circumstances that are actually very specific for Littleanchus crenata. This is a general primer set for nematodes, but it works very well in these circumstances. And so what we're doing is we're using quantitative PCR, calculating the number of gene copies per gram of tissue, and then following how that changes over time. And so when we begin our sampling, we're working with buds, and we're getting copy numbers along the lines of 1 × 10⁻⁹ per gram of tissue. And then once that bud opens up and the leaves emerge, we see this dramatic decline to about 1 × 10⁵. So a 4 order of magnitude drop, probably due to just the expansion of the tissue. So you're dealing with, nematodes packed into a very small space. And of course, as that bud expands, now those nematodes are being distributed over much larger surface areas. So we're seeing a decline probably due to dilution. Then over the course of the season, we see the nematode populations generally track upwards to get back to where they were in the buds originally by sometime in October. Now, this is the next year's leaves, if you will. This is the buds that begin to form in August of 2019. These are the leaves that are present in August of 2019. So these are the leaves, and these are the buds that begin to form as they begin to form that new leaf cohort for the following year. We will not detect the nematode in buds in sort of late July, early August when they're first forming. So they're not present as the buds first form. But by late August, they're present. And then, of course, the population size, the copy numbers of the ribosomal RNA gene gets higher across the season and then sort of plateaus out and stays fairly high through the winter. We can collect buds in February. We can float them, and we can get live nematodes coming out of those buds in February. So they appear to overwinter in the buds. And then you can see here again, as the leaves, as the buds expand and open up, you get that decline again and then it begins to increase. So as someone once said, this is less of a leaf disease and more of a bud disease. So what we think is happening is that the nematodes, once they're established within a tree, are moving from the leaves and they enter the buds starting in late summer through the fall. And they're probably using a water film to move. And so the fall is a good time. You got a lot of thunderstorm activity, you got very dewy mornings, and so you've got a film of water. And the nematodes need a film of water to move. So they will move from the leaves and they begin to enter the buds prior to leaf fall. And then once they're in the buds, they actually begin to feed and damage the tissue. So that damage is already apparent the following spring when the leaves open up. The other interesting thing is that once you see those symptoms in the spring, once that leaf unfurls and you see those symptoms, the symptoms don't change over the course of the season. Don't change over the course of the summer. The symptoms that you see in May are the symptoms that you will have in August. The symptoms do not progress. So this is really more about symptom development in the bud as the nematode feeds in fall and into winter. And then, yeah, you just don't see symptom development or progression over the course of the growing season. It really seems to be taking place mostly in the bud. So properly thought of, this might be more of a bud disease than a leaf disease. But we think that once this organism is established in the tree, that's how it's maintaining itself. It's moving from the leaves and into the buds prior to leaf fall. Now, as I mentioned, we're concerned about American beech, Fagus grandifolia. Fagus grandifolia is the only native species in beech in North America. It's important for wildlife. It produces a lot of hard mass, and so we're concerned about the loss of beech from an ecological perspective because of what that impact might be on wildlife, whether it be small mammals or birds, for example, or even larger mammals like bear and deer. But there are other beech species that are impacted by beech leaf disease. I mentioned Fagus sylvatica, European beech. It is an important ornamental. So here at the Holden Arboretum, when we look at our collection, most of our collection is actually European beech. It's Fagus sylvatica. We have lots of American beech in our forests. We have about 3,000 acres of natural areas, most of which is forest. And so there we're dealing with beech maple forests. But our collection is largely European beech, Fagus sylvatica. And that's the case in a lot of places. It's an ornamental, it's horticulturally important, present in lots of gardens, also affected by beech leaf disease. We also see beech leaf disease on Fagus orientalis, which is native to Asia Minor, also sometimes used as an ornamental. Fagus crenata, I'm mentioning here, again, Japanese beech. We really don't see beech leaf disease on Japanese beech to any great degree. So I'll come back to that. We've done some testing of it. We have some representatives in our collection that don't seem to become symptomatic for beech leaf disease here. And we've done some testing in the greenhouse as well, and it does seem to be somewhat resistant to developing symptoms of beech leaf disease. So this disease developed in Lake County, which is right here. Appropriately, Lake County is on the shore of Lake Erie. And it was first discovered in 2012, as I mentioned, by John Pogachnik in the Cleveland Match Parks. We saw it here in about 2014, but by 2018 it had spread. It had spread to the south and to the east, and also across the lake to Canada. It was present in Ontario. And we kind of thought, well, maybe birds are spreading this because it looks a little bit like a migratory flyway. But by 2021, the spread of beech leaf disease was mostly to the east. And by 2024, it looks like this. And so it's spread very heavily to the east and up into New England. I think there's a current map that shows additional counties that are impacted by beech leaf disease as of 2025. A little bit of spread to the west. So it is in eastern Michigan and western Ohio, the Toledo area, and it's spread a little bit to the south. But for the most part, it's been spreading mostly to the east. So this looks a lot less like a migratory flyway. We have worked with some folks like Danielle Martin at the Forest Service and Chris Latuma and Spencer Parkinson at West Virginia University, Mahal Cantor at Penn State, looking at whether birds could potentially be a vector for the nematode. And so we can detect the nematode on feathers of birds and also in its feces, but we haven't been able to find live nematodes in the feces, for example. So it's possible that birds are a vector, at least for long distance travel. But that, some additional work would need to be done there. It is clear that mites, beetles, and caterpillars can harbor, if you will, the nematode. So Mahal Cantor and his group have found it present on caterpillars and beetles, and it can move meters, 10 meters within a forest, for example, on caterpillars. It's also associated with spider mites. So spider mites sometimes form galls, and Lynn Carter had found some of the mites that she had actually excised from galls. As she excised them, you could actually see that the nematode was entrained in the legs of the mites. So some pretty cool videos of these nematodes coming out of the coming out from the legs of the mites, something like out of an alien movie, quite honestly. So we know that insects can harbor the nematode. And of course, these insects can also move on the wind. The nematode may be able to move on the wind itself. And that could explain that mostly eastward spread and distribution of the nematode to date. So less spread to the west and south, much more spread to the east. And that could be about movement either on the wind or being assisted by insects like mites and caterpillars, for example. Some other questions that have arisen about, well, to what degree can soil be a reservoir of the nematode? We've done a little bit of work looking at leaf litter and soil across the season. And we can find, at least with DNA methods, we can detect the nematode in leaf litter really throughout the year, but we don't see it much in soil. We see it at certain times of the year, but very, very low abundance of the nematode in soil. So we don't, at this point, think or know that it could use soil as a reservoir where it could hang out in soil for a length of time. But we can detect it in soil, but it's very, very low levels of detection. We can detect it in the leaf litter after it falls, probably because the DNA is still in the leaf. Not all of these nematodes don't necessarily get out, and some of them may actually just complete their life cycle in the leaf and die. We don't really have a good sense as to what the life cycle of this nematode is. How many generations take place over the course of a year? It's all very sort of uncertain at this point. So that's what we know really about the nematodes ecology so far, what we know about the disease. But what are the effects on forests really? And so we're concerned about the effects on forests and the ecology of forests. And we see large decline within many of our forested areas. So we see declines in the canopy. We also see declines, especially thinning of the understory. That tends to be the most pronounced. And it may very well be that that's because the nematode likes it cool and a little bit damp and needs a water film to move. And you get those conditions more in the subcanopy area, the understory, than you do in, say, the emergent layer of the forest, where you may have more light and more air circulation. So we see these patterns, but we can also quantify it. In 2009, we set up an experiment across 3 forest sites here in northern Ohio. And the experiment was really to look at the effects of pH change on forest growth and forest microbial communities. And so we set this up in 2009, and we tagged 1,200 trees, many of them beech, and we measure them. We measured diameter breast height every year since 2009. So this became an opportunity really to look at that data and to try to determine how mortality has changed since really the establishment of the nematode and beech leaf disease and to the present. And what you can see here is you can see that prior to beech leaf disease really arriving, this particular forest is in Cuyahoga County, really made itself known in 2016. So from 2011 to 2016, we didn't see a lot of mortality of trees. But after 2016, you can see how very quickly mortality picks up. So we have a lot more beech dying within the forest after the advent of beech leaf disease. Overall, we see about a 30% decline for 30% mortality across that 10-year period of time. So beech leaf disease is causing about a 30% mortality in 10 years. A lot of this mortality is with the younger size classes, things less than 25 centimeters diameter breast height. So a lot of the adult trees, the canopy dominants, that are in the emergent layer, still persist and hang on, but we're seeing that dramatic decline, thinning of the canopy itself. So a lot more light getting into the forest floor, but it's those younger size classes, especially in the understory, that seem to be succumbing and dying fastest. The other thing we looked at was changes in relative growth. So looking at change in diameter breast height from 2011 to 2016, before beech leaf disease really made its presence known and appeared, and between 2017 and 2022. And so you can see these very significant declines in the growth of beech within these forests. So we're seeing dramatic increases in mortality and also dramatic declines in beech growth. Now, Cameron McIntyre published a paper in Frontiers in Forests and Global Change, where he looked at leaf level effects of beech leaf disease, and he was looking at asymptomatic leaves. He was looking at leaves that were banded, so that dark interveinal banding. And then he was looking at those more severely impacted leaves, those what you call those crinkled leaves or those leathery leaves that you see. And one of the things that he observed was significant declines in photosynthesis, A net here on the left, as well as some dramatic sort of increases in stomatoconductance and overall a decline in water use efficiency in those leaves as you go from asymptomatic to the more heavily symptomatic leaves. So the nematode is causing a few changes at the leaf level, right? It's causing really a change in the cell proliferation, the structure of the leaf. It's causing changes, reductions in photosynthesis, increases in water loss, and we see that dark banding as a consequence. And these things, of course, then sort of propagate through the life cycle of the tree and cause those declines and that mortality. And eventually, as I mentioned, the buds will simply abort once the nematode populations become so high that the damage is so severe that they just die, they don't open up. So this is what's going on within the forest and sort of at the leaf level. A lot of the work we've been doing here at Holden is in this resistant seed orchard, as I mentioned, and this is kind of what it looks like in the summertime. There's about 100 trees that we've been working with, and doing a lot of the work looking at nematode populations, looking at leaf communities of both fungi and bacteria, but it also gave us an opportunity to observe these trees up close and personal. And one of the things that we noticed after a number of years was that some of these trees really looked good, despite the fact that they had been affected by beech leaf disease for over a decade. So there in the center, you see a tree that's somewhat heavily impacted by beech leaf disease. These leaves are already becoming sort of that leathery, crinkled leaf, that more advanced symptom stage of beech leaf disease. And so that tree has beech leaf disease fairly extensively. But here on the right, that's a pretty healthy looking tree. There's a little bit of beech leaf disease down here. But for the most part, most of those leaves look really healthy and happy. And so this was our first evidence or suggestion that maybe some of these trees are resistant. And this is a common garden. Many of these trees are 10 to 20 feet apart from one another. And as I mentioned, beech leaf disease has been here for more than a decade at this point. So it spread from Ohio all the way into New England. But yet some of these trees that are just 20 feet away from one another display either very high levels of symptoms or very low levels of symptoms. So that kind of suggested to us that maybe there was some resistance here. Again, this is kind of pulling back to look at not the up close leaf level, but the sort of the whole tree perspective, the canopy level. The tree there on the left, these are taken at different times of year, but these trees, this kind of represents the differences that you might see in the plantation. There on the left, you see a tree that's heavily impacted by beech leaf disease and that dramatic thinning of the canopy. One of the things that we see with that tree there is not only do we see this death of buds, the abortion of the leaves, but what will happen is later on in the year, sort of like in mid to late June, the trees will send out a second flush of leaves. And the second flush of leaves is sparse, but the leaves are very different. They tend to be very, very large. So, way larger than your hand would be. They're enormous leaves. And they tend to be somewhat pale in color. And it seems to be a stress response to the tree losing much of its canopy. And so that's kind of what you see here is these very, very large leaves that are leafing out in June as a consequence of the damage that the nematode is doing and the dramatic thinning of the canopy. But there on the right, that's a pretty healthy looking tree. And they're very, very short distance apart, maybe 20 feet or so. So what's going on here? Are we looking? This is a common garden. The soil conditions aren't very different. Exposure isn't different. What really is going on, and is there possibly resistance? So we began doing some work back in 2021, 2022, where we were collecting scions from some of the forests here, as well as the beech plantation, and then doing grafting. So we were collecting scion and doing grafting really in February of each year. We were doing hot callus grafting, which I won't get into too much, but basically you're putting the graft union against a heated cable to sort of facilitate the taking of that graft. So we did about 264 grafts at that point, of which about 116 survived after 1 year. When they do open up in the greenhouse after grafting, I will tell you that they do have beech leaf disease. They do have beech leaf disease. Those grafts, they do have beech leaf disease. But after they go through the greenhouse for a year and then they drop the leaves, the following year when they open up, they no longer have beech leaf disease. We think the reason for this when we do greenhouse work is that the trees are pot watered, right? So the leaves never get wet. They're only watered in the pot. So as a consequence, even though the nematode could get into the leaf, in case of the graft, it's probably in the buds that we're bringing them in. But in case of some of our experimental manipulations, we're adding the nematode. But because the leaves are never wet, the nematode can't get out of the leaf. It needs that water film to be able to get into the bud and continue that cycle. So if we're pot watering in the greenhouse, they're basically trapped in the leaf and they can't get out. So the following year after they let out, we see no signs of beech leaf disease. So 116 grafts survived. They did show some signs of beech leaf disease, but after they went through another growth cycle, they had no signs of beech leaf disease or nematodes. And so they became a good test for us to be able to do some challenges. So I'm going to call this a challenge test. This is the same way we determined that the nematode was causing the symptoms of beech leaf disease. So as I mentioned, we collect the nematodes, we float them in water, we collect leaves and buds, we float them in water, the nematodes come out, and then we collect those nematodes. We can count them, make sure we have an equal number of nematodes, live nematodes going onto each bud, and then we inoculate buds as you can see here. So basically what we're doing is we're taking an asymptomatic individual, we're placing a small amount of Kim-wipe on the bud and just wetting it slightly so it adheres. And then with a pipetter, we take an aliquot of water that contains our live nematode and we very carefully apply it to the surface of the bud underneath that Kim-wipe. And then we wrap the bud in parafilm to keep the moisture in and give those nematodes an opportunity to colonize the bud. We have in the past injured the bud slightly by poking through the bud scales with a dissecting needle. That little bit of damage can facilitate entry of the nematodes into the buds under some circumstances. We typically use about 100 nematodes per bud inoculation. This is what we've used in the past to demonstrate that the nematode doesn't in fact cause beech leaf disease, but we're also using it as what you could call our minimum effective dose for some of our tests looking at differences in symptom development between American, European, and Japanese beech, which I'll show you. So this is what we did in our challenge tests to really kind of identify whether some of these trees, these American beech, were potentially resistant to symptom development and also differences among species of beech. So we've just kind of finished up some of this work. And what I'll say is that we do have evidence that the genotype of the tree significantly is related to the symptom development of beech leaf disease. So under controlled conditions, genotype is significantly related to symptom development. So that suggests that there could be some genetic basis for resistance. And so this is going to require a lot more study and a lot more work. But if there's a hopeful story here, it could be that some American beech trees are resistant. We do see this within some of our forested sites as well. A lot of the trees are declining, but then you'll come across a beech tree in the midst of that's very, very healthy and doesn't seem to have much beech leaf disease. So there is some evidence for resistance here. One of the recommendations that we've been providing to homeowners or property owners is if the trees are healthy, don't try to proactively cut them down. You could have resistant trees. If you proactively cut them down, then you're removing a tree that could be resistant. And if the tree is heavily impacted and causing a hazard, of course, or a potential hazard, then you should remove it. If the tree is healthy, looks good, there's no reason to proactively cut it down because there could be some resistance, either genetic resistance or resistance based on environmental conditions. These two trees here just kind of illustrate again the differences in symptom development. These were tested within our greenhouse study. Tree on the left there is one of our control trees that has high levels of beech leaf disease symptom in the field and does again in the greenhouse. And the tree here, 57T, shows very little signs of beech leaf disease in the field and is indicative of what we're seeing under greenhouse conditions. We also looked at differences in species of beech and symptom development. So looking at Fagus grandifolia, American beech, Fagus sylvatica there in the middle, which is European beech, and Fagus crenata, which is Japanese beech there on the right. And as you can see, all of these trees, the buds received 100 nematodes in fall, waited till the following spring for a symptom development. You can see American beech, fairly high distribution here, but we're looking at about 50% or so of the leaf area being symptomatic, a little less in European beech, but no significant differences there. But once we get into Japanese beech, we're getting maybe 5% symptom development. So identical conditions, identical number of nematodes, but significantly lowered symptom development with Japanese beech. And this could be because this nematode evolved with Japanese beech, that Japanese beech has some sort of inherent resistance. What that resistance might be, whether it's chemical or whether it's anatomical, is unclear. But it seems as though Japanese beech does have some resistance to this nematode. And these pictures kind of highlight those differences visually. So this is European beech on the left. So looking at these dark banded areas. So the bud was down here. All of these leaves emerged from the same bud that was inoculated. And you can see all of the banding on all of the leaves that emerged from that bud. Fairly significant amount of banding. This is Japanese beech on the right. All of these leaves emerge from the same bud, and this is really the only spot where we see that dark banding. So some dramatic visual differences and the degree of symptom development between these two different species of beech. So in a way, I think that's kind of hopeful in terms of what we see within our forests. We do see a lot of mortality. We see declines in beech, but there may be the possibility of resistance, and it does require a lot more study. But we do see some evidence for the existence of resistance. We see differences among the species. But what do we do really from a landscape perspective? We receive lots of calls from the homeowners and property owners. What do we do with our beech tree? And so we've worked with Bartlett Tree on pesticide applications in the past, and Bartlett has done some work looking at different pesticides and found that fluoropyrum, which I'll show some slides on, can control the nematode and beech leaf disease. Davy Tree has done some work with polyphosphite 30 in soil applications that shows some promise. We're currently working with Quest products, looking at different types of polyphosphite applications and what might do the best in terms of controlling beech leaf disease, and doing that again at our beech bark disease resistant plantation. And also we've looked at structural pruning. We've looked at structural pruning and whether it can control beech leaf disease, because as I mentioned, the nematode kind of needs a water film to move around. So if you can open up the canopy and you can increase air and light circulation, you can increase the dryness of the buds and the leaves, and maybe that prevents the nematode from moving around. So I'm going to show you the structural pruning results first. What we're doing here is we're scoring the trees on a scale of 0 to 5, with 5 being more than 75% of the canopy affected by symptoms, 0, of course, having no symptoms. So if the change in score goes up, if it's positive, that means there's an increase in symptom development. If it goes down, it means there's a decrease in symptom development. So we're seeing when we prune trees, 1 year after pruning, we're seeing a significant decline in symptom presence on trees. And this work was done on European beech, so cultivars of Fagus sylvatica within our collection. These are large trees, so many of them are 30, 40, 50 feet tall. So we had an arborist do this for us. So we see that significant decline after a year. After 2 years, 2 years post pruning, we don't see a significant difference anymore. We see that there's a lot more variability here within the prune trees, but we see no real difference here between control and prune trees. So it's just that the pruning effect, the benefit of pruning, may wear off over time. Some of this may also be due to the year itself. So you can imagine that a very wet fall, late summer, could be more conducive to the nematode being able to move around and get into those buds prior to leaf fall. So some of those scores will change potentially based upon the weather conditions. But pruning does seem to potentially be one way of controlling or reducing symptoms. This is showing the scores really for the individual trees. So we had 5 trees that we scored as control trees and 5 that were pruned by our arborist team. And in all of the 5 trees that were pruned, we saw declines in scores. So So symptom development actually was reduced. The scores improved, if you will, where the trees were pruned. In control trees, we either saw effectively no change, or we saw some slight increases in the disease rating. So some increases in symptom development in control trees from one year to the next. But the prune trees generally had reduced symptoms. So that suggests that pruning, structural pruning in this case, can reduce symptom development. So that can be useful on the landscape level. As I mentioned, pesticide control is one way to go. Bartlett tree has done a fair amount of work looking at different pesticides. Indemnify is the one that seems to have the best results. Indemnify is here in the middle, and that's fluoropyrum. So where Indemnify was applied, the samples that they took, they had an absence of nematode in all of the samples after treatment with Indemnify or fluoropyrum. So indicating that this can be used to kill the nematode and potentially control beech leaf disease. Davy tree has done work with polyphosphite 30. This is courtesy of the Cleveland Metro Park. Stan Volk was involved with this project, as well as Connie Hausman there. And they treated trees for 5 years. And so they saw significant reductions in nematodes, fewer symptoms, and a fuller canopy in areas that received polyphosphite 30. And that's the picture here in the middle, as compared to plots as shown on the right that were the control plots that did not receive polyphosphite 30. So there's some evidence that polyphosphite 30 will reduce the number of nematodes present and reduce symptoms of beech leaf disease and allow for the trees to improve their health and growth. So as I mentioned, we're doing some work here now looking at different formulations of polyphosphite 30. We've applied soil drenches, similar to what I believe that the Metro Parks did with Davy Tree, as well as basobaric applications and some granular applications of polyphosphite 30 to see which actually might give best control of beech leaf disease. Some other work that we've done, and I'll just mention this because this kind of relates really more towards the forest ecology pieces. We know that the trees are declining in our forests. We know that growth is reduced. We know mortality is increased. What does this mean for forest composition? So anecdotally, I can tell you that when the canopy is thinning here, we see the succession taking place. And in some cases, that's not all that bad. We're seeing tulip poplar, sugar maple coming in. We're losing, a hard mass species, but at least sometimes we see natives like tulip poplar and sugar maple coming in. But in other places, we see a lot of buckthorn, a lot of multiflora rose coming into these communities. And so there's a fear that forest composition can change. So Katie Stubel, my colleague, and Brianna Shepard, our research specialist, set up 113 plots across Pennsylvania, Ohio, and West Virginia. And in these plots, we are measuring disease severity, understory cover, taking soil samples, and actually working on sort of quantifying exactly what we might expect to happen as these beech trees decline. What can we expect in terms of forest community responses? So that's some more to come. We've done this for the last couple of years. And so I'm just going to kind of wrap up with some potential future questions, leave some time for questions for all of you if you have some. But some things we're working on right now is really weather effects on beech leaf disease symptoms. We do see changes in the symptoms from one year to the next. So 1 year, the symptoms will dramatically increase in terms of their extent and the extent of the canopy affected. And in another year, it seems to improve. And we can quantify that as well. We do measure these trees at our plantation every year. We've been doing it since 2018, and we can tell you that symptoms go up and down across the years. So what is the effect of weather, for example, on beech leaf disease symptoms? We're also interested in this question of soil chemistry and nutrients, especially in terms of whether microsite differences within a forest could potentially be affecting symptom development. And as I mentioned, some trees like Japanese beech show very few signs of beech leaf disease. What is the mechanism of that resistance? We're seeing some potential effect of genotype on symptom development in American beech. We don't know why that's the case, but that requires additional study. And one thing that I can't give you is a conservation recommendation. So I can tell you if you're a homeowner, if you're a gardener, if you in charge of a park system, that there are different types of chemical applications you can use. Structural pruning may help. But from a forest management perspective, what do we do? And so that's something that we can't provide recommendations on, but something that we're looking to study in the future. And with that, I'm just going to pause for some acknowledgments. Jennifer Cook and the U.S. Forest Service, the major collaborators on all this work. A number of people in my lab have contributed this over time. We've received funding from the Forest Service, from Holden Forests Gardens, the Great Lakes Restoration Initiative, and also the Nature Conservancy through the Tree Species in Peril program. And with that, I think we have maybe 10 minutes or so for questions.
We sure do. Do we have any questions for David? All right. I am not seeing anything pop up.
That means I did a great
Yes.
job answering the
Yes, you
questions in
did an
advance.
excellent job. Let's see. I do see, can you reiterate your prediction on the spread to the West Coast?
Yeah, I think, some of it does really look like it's being sort of carried on the wind to the east. And so it could be that nematode's just moving on its own. And so the movement to the west has been much slower. We're seeing it in Western Ohio near Toledo. We have been monitoring some places like Houston Woods, which is on the Indiana border, but we haven't really seen any symptom development in those more western counties. So some of this could be due to, like I said, the prevailing winds. Some of it could also be due to just the prevalence of beech. Sort of, I think as we move west, the dominance of beech or the presence of beech kind of thins. And so the lack of extensive beech coverage could be, hindering the nematode spread to the west and to the south potentially. But yeah, we do see it in eastern Michigan at this point, but that's about as far as west as it's gotten.
All right. Our next question is with survival at negative 80 degrees, do you think there's any northern limit to the disease within beech range?
I'm not sure. I mean, we see a lot of spread into Canada and up into New England, so it gets very cold there. Minus 80 Celsius is, that's crazy cold. So that's about minus 80 Fahrenheit. So it's possible that it could be limited in terms of cold temperatures, but it's something that we have really yet to determine, and it would require some additional work. We don't see it moving too much to the south, whether that's because of the prevailing winds or whether that's a question of dryness. That's another thing that we consider is what is the effect of rainfall and precipitation patterns. And maybe that's one reason why we're not seeing movement to the south quite as much. But yeah, it takes a lot of really cold temperatures to kill them.
All right. Our next question, would you recommend for planting purposes that the planting of beech be reduced in urban areas?
Yes. So people ask us whether they should plant beech, and we say no. Japanese beech does appear resistant, but it's very difficult to get Japanese beech. Japanese beech was used in the bonsai trade. So there's a period of time in which you could actually acquire Japanese beech, but we've tried to purchase Japanese beech. It's very difficult to get it anymore. I think people have stopped selling it, but it's not typically considered to be a landscape specimen. Most of the beech people want to plant in their yards or gardens or parks is Fagus sylvatica, and we're just telling people don't plant it. Select another tree species.
All right. I see a question from me there about ISA CEUs. I was not able to get CEUs for this particular session. You can always try self-reporting. And if they come and ask, I can verify that you were present by the attendance that Zoom provides. So I wouldn't worry about that. I do plan to try to get credits available for people with the last two sessions this semester. And then I see one more question here. Could you reiterate whether there's a treatment option or if it's only a preventable disease?
I think it's a treatment option. I think the things like fluoropyrin will treat it, kill the nematode, but of course that it's not going to eliminate entirely. So the tree can become reinfested, and so it could require additional application. With polyphosphite 30, Again, it does require reapplication. So depending upon the form, if you're using, say, the soil drench, it might require repeated application. But that's kind of what we're trying to test right now. How much control do you get with the different formulations? What would be the best recommendation in terms of reducing symptom development? It may be for landscape specimens, you want a combination of things. Maybe you want to do structural pruning to open up that canopy, but then polyphosphite 30 application might also further help, especially for, like I said, we're telling people don't plant it, but you can imagine that you could have a really beautiful specimen of European beech in your garden, in your park, and you really want to preserve it. And right now, it looks like polyphosphite and some of these other chemical treatments, coupled with structural pruning, might be able to help keep that tree alive and reasonably healthy.
And then I see someone asking, can we provide a detailed outline of the presentation for CEU reporting? Um, I wish that I could. Unfortunately, there's just this issue with having enough time to be able to put such a thing together. However, this is being recorded and we will post it on YouTube and you can refer back to it. You'll get a link to the recording as soon as it's posted. However, if you're worried about CEU reporting, I have never seen anybody have any issues reporting this particular program series with CEU stuff. This is a long-running series. People at ISA know it exists. If you run into any problems, just reach out to me. I can contact people over there and we can get you set up. All right, I am not seeing any other questions, and it is almost 2 o'clock. So I'm just going to say if you have more, feel free to send me questions. I can share them with our presenter today, who did an excellent job, by the way. That was really, really interesting, and I'm happy that we got to
Thank
have
you.
that. But with that, I want everybody to have a great weekend, and hopefully we'll see you at the next one. Take care, everyone.
Have a good day.